Hello everyone, today, it is my honor to introduce QSCNAS, a platform for quorum sensing and quenching bacteria analysis in global wastewater treatment plants. As an important infrastructure of modern urban society, wastewater treatment plants play a key role in controlling pollutant discharge. In this process, the activated sludge and its derivative improvement process have become the most widely used method for treating sewage. It is important to note that performance of the activated sludge is influenced by microbial interactions. The communication process between bacteria, known as quorum sensing, has received a lot of attention in recent years for its ability to assist in multi-population behavior. However, understanding the behavior of population communication rate and the role of different signal types in wastewater treatment systems remains a serious challenge. Considering the importance of bacterial communication in the biological treatment of sewage, this study aims to construct a bacterial database and a communication network based on signal molecules. We first developed a local database to identify QS and QQ bacteria based on signaling molecule secretion, acceptance quenching. Using more than 1,000 activated sludge samples collected and sequenced by the Global Water Systems Microbiome Consortium, we identified potential QS and QQ bacteria in activated sludge and built an online platform for researchers to search and analyze microbial communication-based interactions in activated sludge. The results showed that the bacteria related to CDGMP and AHLs dominated the communication system. Taxonomic analysis has shown that proteobacteria accounts for more than 50% of the relative abundance in various communication systems and contains almost all functions related to signal generation and quenching activities. At the same time, some bacteria with important functions, such as Candidatus accumulobacter and Candidatus microfrix, are not only abundant in the global WWTPs, but also associated with multiple QS signaling languages. The results on the single-mode network showed that the number of receptors in AHL-type QS bacteria is much larger than the number of synthesizers, which is in stark contrast to DCEF and AI2 QS bacteria. DCEF-associated bacteria outperformed AHL and AI2 in abundance, frequency and coefficient of variation, indicating their importance and potential contribution in creating social benefits. At the same time, it was found that the bacteria participating in QS communication had strong positive correlation, and there were a considerable number of keynotes. By constructing abundance co-occurrence networks, a high degree of cooperative behavior among these bacteria was observed. In particular, the network parameters of these multilevel interspecific communication bacteria in the QS network were significantly positively correlated with those in the co-occurrence network, as well as their relative abundance and coefficient of variation in the global WWTPs. At the same time, the results showed that the Nitrospiralis and Rhodobacter had high QS connectivity in the community, which may serve as the main bridge of QS communication network and promote the realization of function. The abundance of these QS bacteria was also significantly positively correlated with their performance. In this study, through the collection and analysis of more than 1,000 activated sludge samples worldwide, we built QS and QQ bacterial databases in activated sludge to form an online network analysis platform. QS bacteria affected the function of wastewater treatment plant through direct and indirect effects and maintained a high positive correlation with the performance. According to their own needs, readers can use the browsing, classification search, network construction and functional analysis and other functions.